First, since migraine is a polygenic trait resulting from the additive effects of hundreds or even thousands of genetic variants in many genes and other environmental contributions, a GWAS is performed to identify regions of the genome that are associated with a given phenotype, which in our case is migraine. To perform the GWAS, thousands of individuals with and without migraine are recruited and classified as cases and controls. Then their DNA is genotyped, and we look at the frequency of genetic variants across the entire genome. If a genetic variant is more common among individuals who have the disease than for those who do not have the disease, then that means that the genetic variant is somehow associated with migraine. And thanks to GWAS studies in 2016, 38 single nucleotide polymorphisms across the entire genome were associated with a higher risk for migraine. And more recently this year, an unpublished preprint identified 123 SNPs associated with a higher risk for migraine. All of these SNPs were identified through Genomewide association studies. And later on, we can use GWAS results to estimate genetic correlations between different phenotypes. Genetic correlations are estimated using LDS co-regression and indicate the extent to which two phenotypes share the same genetic etiology. These genetic correlations are meaningful because they are robust to confounding due to Mendel's laws. That means that the estimates of a genetic correlation are unlikely to be biased by external factors due to Mendel's law of segregation and of independent assortment. However, the genetic correlation only allows us to know to what extent are two complex traits sharing the same etiology. But we still need to assess potential causal effects between these traits, and that's when the latent causal viral method comes in. This method, also known as the LCV method, creates an inferred causal pathway which mediates the genetic correlation between trait A and trait B through a latent variable L. And this latent variable L is assumed to be the causal constituent mediating the genetic correlation between both traits. The advantages of this method include that it is able to handle sample overlap, it is less susceptible to confounding by horizontal pleiotropic effects, and it uses aggregated information throughout the entire genome to increase statistical power. In our study, we used GWAS data for migraine and 1,504 complex traits to estimate genetic correlations using LDS co-regression. And then we assessed potential causal associations using the latent causal variable method. We identified 517 genetic correlations with migraine, of which 17 were explained by a potential causal association. Overall, 3 phenotypes were potential causal outcomes of migraine, while 14 traits were potential causal determinants of migraine. We broadly classified phenotypes that have a causal association with migraine into 4 different categories. These included cardiovascular phenotypes, abdominal discomfort and pain, emotional distress, and workplace environments. In particular, hypertension and blood clot formations were causally associated with an increased migraine risk, possibly through vasoconstriction and platelet clumping. We also observed that sources of abdominal pain and discomfort might influence a higher risk for migraine. Another of our findings was that occupational and environmental factors, such as working with paints, thinner, or glues, and being exposed to a diesel exhaust, were causally associated with a higher migraine risk. And finally, psychiatric-related phenotypes, including stressful life events, increased risk for migraine, while, for example, ever feeling unenthusiastic or disinterested for a whole week, which is a phenotype related to the psychological well-being of individuals, was a potential outcome of migraine. Things to take away from these findings are that our results suggest that there is a potential vascular component to migraine, and we particularly highlight the role of vasoconstriction and platelet clumping. And we note that stressful life events and occupational variables potentially influence a higher risk for migraine, and migraine could impact the psychological well-being of individuals. I believe that one of the most important things to take away from this study is that in order to establish causal associations between complex traits, it is critical to triangulate results with multiple study designs, of which at least one should be ideally an interventional study, such as a randomized controlled trial. However, as I mentioned earlier, interventional studies can be expensive, time-consuming, and even unethical to perform. Therefore, identifying potential causal associations using statistical methods in genetic epidemiology could be the best informative option available in some cases. And our results contribute to this by generating testable hypotheses that can be confirmed by future studies. And we also provide novel insights into a relationship between migraine and the overall health.